How's everyone doing? If you haven't jumped into the Menti, jump in, let me know. Let me check in on everyone. I'm seeing a distinct lack of people in the green zone. Some pleasantness, unpleasantness, lower energy, high energy. So still kind of, what is that one? Let me see. Exhausted, okay. Miserable, sure. Morose, concerned, stressed, okay, cool. Some pleasant, some at ease, some coming in. So still getting a spread, maybe a little bit more unpleasantness. Is there any general things going on? Still have midterms or extra term tests coming up. I'm seeing some head nodding, assignments due, grades getting released. A lot of things kind of just stressing you out, so it's understandable. Just take that moment to check in on yourself. I know for myself, walking in here probably a little bit more low energy, but kind of getting into that yellow zone now, a little bit more high energy. I'm going to go lively today. High energy, a little bit pleasant. But yeah, I think that's where I am. But again, go through this exercise, take some time, check in on yourself, see where you're at and see where you need to be is generally a good exercise just to bring awareness to yourself. Well, thank you for sharing. We're going to get started today on the second, not really the second half, but kind of the second half of the course. So today we're going to really start diving into metabolism. Dr. Patterson, if you remember before reading week, started that discussion on metabolism, thinking about the thermodynamics and how energy and thermodynamics contributes to the setup of these metabolic pathways, coupling enzymes together. This week we're going to start looking at some of these metabolic pathways in more detail, and specifically looking at energy metabolism. Now, while the second half of the course, and the final exam is going to focus on the second half of the course, not going to be cumulative, this is going to be building on those concepts we talked about. We're going to see protein structure. We'll continue to see protein structure and how that contributes to protein function. We're going to start looking at pH buffers, structures, functional groups. All of that contributes to it. We're going to see enzymes. We're going to see transporters, membrane proteins. All those concepts we talked about before reading week are all going to come back now as we start to look at this metabolic pathway and how cells derive energy from energy-producing macronutrients, in particular carbohydrates. Well, for the first half, carbohydrates. Later on, looking at fatty acids as well. So what I want you to be able to focus on at the end of this lecture is to be able to explain the importance of carbohydrate structure and how that contributes to metabolic processes, describe how the functional groups are organized in a carbohydrate, and how that organization contributes to the complexity and why we need a number of enzymes or a whole suite of enzymes in order to break those down to derive energy from those molecules. I want you to be able to differentiate between the synthesis and breakdown of those carbohydrates. So how do we make complex carbohydrates, and then how do we break them down? And then finally, at the end of this lecture, we're going to look into how carbohydrates are transported within the body and seeing how those proteins contribute to the movement of glucose down this concentration gradient to get that glucose into the cells so that they can use them for energy. On Thursday, we're going to start taking a look at glycolysis. So it's all kind of building towards that point. So how do we get glucose from the food you eat into the cells where they can use it for energy? But I want to start with a case study. Show of hands, how many people are thinking med school, dentistry, optometry, pharmacy, some sort of professional program? Show of hands. Nursing. So I previously taught in a nursing program, and a lot of times in health professional education, we'll use a case study to kind of put everything into context. So I want to bring this more physiological case study into our classroom, and I want you to think about this as we move through this lecture and how the concepts we talk about relate to this case study. So this case study here, RP, is a 12-year-old who has recently been diagnosed with type 1 diabetes mellitus. So type 1 diabetes mellitus is an autoimmune disease in which the immune system targets the beta cells of the pancreas. These are the cells that produce insulin and destroy them. Eventually, that person can no longer produce insulin, and this has significant impact on a person's ability to regulate blood glucose levels. We're going to see how insulin contributes to the regulation of blood glucose. You've probably seen that in some other courses as well. So this is going to be some concepts that are going to keep coming up. Now, some extra context for this case. Recent blood work indicates that RP has a hemoglobin A1C level of 12.1%, indicating a higher than normal blood glucose level over the past 3 months. Now remember, we've looked at hemoglobin, we looked at the structure of hemoglobin. Now we're seeing how hemoglobin can be used as a diagnostic tool. So keep in mind what that 12.1% might be and how that might indicate or help us understand what's happening with the regulation of blood glucose levels. And then secondly, RP has also experienced recent fainting episodes following treatment with insulin. So exogenous insulin or a dose of insulin to help them regulate the blood glucose levels. This suggests periods of hypoglycemia, hypo meaning low, glycemia, blood glucose, so low blood glucose following treatment. So keep this in mind, those 3 points. Hemoglobin A1C levels, 12.1%, a lack of insulin, and hypoglycemia following insulin treatment. So as we move through this lecture, we're going to come back to this case study a couple times to examine and put our understanding into the context. But now I want to just come back to you, kind of setting the foundation. Where have you seen glucose in the context? Okay. Where have you seen glucose in the context of biology and biochemistry? And what have you seen it used for? So take a second, jump into the Menti, and let me know. Nope. Where have you seen glucose in the context of biology and biochemistry? What have you seen it used for? All right, so let's see what's happening here. We got main ones, glycolysis. Absolutely, we're going to see glycolysis later on Thursday. ATP, so generating energy. Cellular respiration, we're going to talk about that a bit more next week. Photosynthesis, not going to talk about it in this course, but this is going to be how plants are able to take energy from the sun and take basic atoms and nutrients, carbon dioxide, water, and make those carbohydrates or those glucose within the cellular or plant systems. As an energy source. GLUT transporters, GLUT2, GLUT4, we're going to see those again later on. Krebs cycle. Anything else? See diabetes, membranes, GLP-1. Yep, we're going to see that with your assignment, or you have already seen that with your assignment. Glycogen storage, fed and fasted state. Absolutely. Brain, absolutely. So brain is an interesting piece and an important point when we start thinking about carbohydrates within the body. So, as I kind of were highlighted in some of those responses that came in, carbohydrates are one of the energy-producing macronutrients found in the diet. Carbohydrates, proteins, and fats. This is where we derive our energy, and you're going to see them highlighted on the nutritional label where we derive our energy from. Under carbohydrates, you'll also see fiber, which are going to be non-digestible carbohydrates. So it's carbohydrates we can't digest, so they just move through our digestive system and they're excreted in our feces. Simple sugars, as well as a nutrient of interest, a concern, so it's also highlighted on the nutritional label. But like fats and proteins, you get energy out of carbohydrates. On average, you derive about 4 kilocalories or 4 calories per gram for a carbohydrate. So you can use this to calculate the amount of calories you get per gram of carbohydrate you consume. Now, the recommended dietary intake for glucose is about 130 grams per day, irrespective of body shape, body size, person, sex, gender, anything. This is a really important number. This number is derived from how much glucose your brain needs to function. So 130 grams per day is the lowest amount of glucose that your brain needs to function per day effectively. We calculate that out. That means times 130 grams is going to equal to about 520 kilocalories. That is the amount of energy your brain needs to function. Now, it's important when we think about glucose, and we're going to see when we think about glucose transport, the brain relies on glucose for energy. So we need to be able to get glucose from our digestive system through our blood, cross the blood-brain barrier, to our neurons to function effectively. Our brains cannot use fats for energy effectively because fats cannot cross that blood-brain barrier effectively. So they rely almost entirely on glucose and some other molecules we're going to talk about later in the course as well. So glucose and carbohydrates are critical for the functioning and maintaining homeostasis in the body. As a note, we see fats, proteins, and carbohydrates, and we're not going to go into this course. What happens to all the nucleotides you consume? You're consuming lots of DNA. We don't list it on the nutritional label, but it has to be going somewhere. I'm going to leave that one with you. If you're interested, go and search it up. See what happens to all the DNA you consume. So looking at carbohydrates, there are some familiar... There are some familiar carbohydrates you're going to see within your diet. We have glucose and fructose. These are monosaccharides that come together, forming a glycosidic bond. We're going to talk about this in a little bit later, forming sucrose. So this is a common disaccharide we're going to see, or you're going to see when you get from fruits. They're going to give that sweet taste that we're going to consume. We also see some pentoses. So these are 5 carbon rings of 5 carbon rings for carbohydrates, or ribose and deoxyribose. You can see within your DNA and RNA. We also have glucose that's going to be part of a post-translational modification. So we have N-link glycosylation or O-link glycosylation, where a glucose molecule can be added onto specific amino acids. These are post-translational modifications we've spoken about. But all these carbohydrates have the basic general formula, CH2O times the number of N's. This is literally a hydrated carbon, a carbon with a hydrogen, a hydrated carbon or a carbohydrate. Every carbohydrate is going to have the same general molecular formula. So this is one of the underlying characteristics of a carbohydrate. In addition, they have in their linear form, you're going to see they're going to have an aldehyde or a ketone group, so a carbonyl group, and a number of hydroxyl groups. This gives a carbohydrate its general chemical structure. It is important for how it functions and how we use it within the body. So I have a question for you now, jumping back onto Menti. Sucrose and saccharin both bind to G protein coupled receptors, but saccharin is 100 times sweeter. What do you think could account for this difference? Is it that saccharin binds more tightly to the G protein coupled receptors? Saccharin is metabolized faster than sucrose? Saccharin produces more energy? Or sucrose breaks down glucose to fructose, with glucose and fructose, which are less sweet? What do you believe? Why do you think saccharin, which is non-nutritional sweetener, that's sometimes added to different foods to stimulate sweetness, but you don't derive any caloric intake from it? Why do you think that is? Seeing some responses coming in. So, slight majority going with saccharin's metabolized faster. A few people are slight majority saying that saccharin binds more tightly to the G protein coupled receptors. Second choice, probably saccharin's metabolized faster. But in this case, it's all about binding to that receptor. So think about perception, think about cellular intake, everything is about binding to receptors. So we've seen some receptors in the context of our course. We've seen a G protein coupled receptor when you're thinking about your assignment and GLP-1 receptor. We've seen them a few times within the course. These receptors are on the surface of proteins and allow something on the outside to have an impact on the inside of the cell. Looking at the structure of sucrose and saccharin, very polar, lots of carbonyl groups, nitrogens on saccharin, lots of hydroxyl groups. This can cross the plasma membrane. This means it needs to bind to a receptor. When it binds to the receptor, it's going to initiate a cascade, a signaling cascade that we can perceive in our brain as a taste. And in particular, I added this to the slides. It's not in your deck. It's not testable, just for context. So all of those taste receptors, and here is specifically the sweet taste receptors, are G protein coupled receptors. They bind to these molecules, initiating that signaling cascade inside the cell. So similar to what you see with GLP-1, you've seen in your draft, similar to what we saw with the beta-2 androgenic receptor, these help connect the outside to the inside. When these molecules bind to the receptor, so we're seeing sucrose, saccharin, aspartame, they're going to initiate that signaling cascade by binding to that receptor. If they bind to a receptor more tightly, they can initiate a much stronger cascade, giving you a much stronger sensation of sweetness. Even if saccharin's metabolized much faster, it's actually going to make it disappear very quickly, limiting the amount of sweetness that you're going to be able to detect. If it's not around, it's not going to initiate that sweet taste. So it's really about it binding to that G protein coupled receptor. You can think of this as its KD, its dissociation constant. A much lower dissociation constant means it's going to bind much tighter to that receptor. So thinking about monosaccharides and these carbohydrates, the simplest forms we're going to see are usually trioses. So these are going to be trioses that have 3 carbons. So carbon 1, 2, 3, forming a triose. And that suffix of os, OSC, is usually going to indicate a carbohydrate. Just like A's, ASC, indicated an enzyme. When you're thinking about a nomenclature for carbohydrates, whenever you see OSC, you should be thinking carbohydrate. Generally, this is going to divide our carbohydrates into 2 types, an aldose, so having that carbonyl as an aldehyde group, or a ketose, having that carbonyl as a ketone group. This structure and the distribution of these functional groups generates some chiral centers. These chiral centers are very important for the complexity of carbohydrate structure. A triose, the simplest carbohydrate, has 1 chiral center. This means it's going to have an isomer, just like with our amino acids, L and D. However, as the size of those carbohydrates increase, the number of chiral centers also increase, dramatically increasing the number of isomers and the number of different configurations you're going to have for that carbohydrate. Now, I do want to take a second to pause to talk about numbering. Numbering in biochemistry, or numbering for carbohydrates, we usually like some steadfast rules. So some of the easiest rules, or the best rule that I can imagine or can tell you, is to start with the most oxidized side in its linear form and start to count down. So being able to picture these carbohydrates in their linear structure and counting from that most oxidized, or the side that has the carbonyl or the aldehyde group, is going to give you the easiest way to number those carbohydrates. And this will be a skill that you're going to be expected to know for the final exam. So when we start to number these, we can start looking at the number of carbons. So triose has 3 carbons, tetrose has 4 carbons, pentose has 5 carbons, hexose has 6 carbons. Numbering from the carbonyl group, number from the most oxidized group, or the carbonyl group, the least oxidized, in its linear form gives us our numbering system. So carbonyl group at the top, 1, 2, 3, we can number our way down. The uniqueness here is when we start thinking about the difference between an aldehyde and a ketone, that ketone does not have that carbonyl group at the terminal carbon. So we still organize it in that linear form. We just place the carbonyl group at the top, or closest to the top, and start counting down. But in this case, number 1 will still be that methyl, or the methyl hydroxide, 2, 3, and then 4, numbering its way down. So follow the same rule, line it up straight, top to bottom, put the carbonyl group at the highest point, and start counting your way down. Now, because of that configuration, because of the number of chiral centers, we have a lot of different isomers. In an amino acid, you have 1 chiral center, that means you have your L and D conformation. For a carbohydrate, because you have multiple carbon centers, you have multiple different isomers. And each one of these is going to be labeled as L or D. We are going to define them as lowercase L or D for the ones closest to the carbonyl group. When we get to the chiral center farthest away, this is what's going to distinguish something as being a capital L or capital D isomer. So here we see D-glucose based on the orientation of that last chiral center. Yes, L and D is going to be assigned based on the chiral center furthest away. But this does increase the complexity of carbohydrates dramatically because each one of these has an isomer. Looking at this branch structure, or this kind of family tree of carbohydrates, as we move down, we add chiral centers, so we increase the complexity. And we can calculate the number of potential isomers for a carbohydrate based on the number of chiral centers. Using the formula 2 to the n, where n equals the number of chiral centers, we can figure out how many different isomers there are. So looking at, say, the aldotetroses, we have 2 chiral centers, so 2 to the 2 is equal to 2 times 2, which is equal to 4. However, we only see 2 here. The reason is that this figure is only showing the D isomers. There's a whole nother replicate of this as L isomers as well. So this gives us 4 total isomers for the aldotetroses. So the complexity increases dramatically as we get down to the aldohexoses or aldoseptoses with 7 potential isomers. You may have 32 to 40 different potential conformations or configurations for those functional groups. This makes them really complex, and their structure and the association of or where those functional groups are is critical to how our enzymes are going to interact with these molecules. So, question for you then, how many isomers of D-ribose are there? Is it 2, 4, 8, 16? How many isomers of D-ribose are there? Majority going with 8, which would be correct. Let's take a look at that. So looking at D-ribose, we have 3 chiral centers, 1, 2, 3. That's going to mean the number of isomers potentially are 2 to the 3, which is going to equal to 2 times 2 times 2, which is going to equal to 8. So looking at the number of chiral centers, you can determine the number of potential isomers for that molecule. Now this means that the number of structures for sugars or carbohydrates are very diverse. They are going to have the same basic chemical formula, CH2O, times the number of carbons that they have, or hydrated carbon. They're going to be able to be configured in many different ways in order to generate the same chemical structure. And then to add some complexity to it, there's a different conformation where it can go from its linear form to its cyclic form that you might normally see associated with glucose. This ring structure adds another potential chiral center, creating another isomer. So that cyclicization reaction is due in part to the reactivity of the aldehyde group or that ketone group. That ketone group or aldehyde group is very prone to a nucleophilic attack. Something attacking it, like a hydroxyl group, attacking that ketone group to generate that ring structure. But carbohydrates have lots of hydroxyl groups in them. So it has a built-in functional group to attack that carbonyl group to form this cyclic ring structure. Now the generation of this ring generates a ring with an oxygen in it through the formation of this hemiacetyl or this hemiketyl group. Now this association or this similarity means that we name these groups or these ring structures as furans or furanoses or pyranoses based on the formation of the ring. So furanose, that os suffix, or pyranose. This new ring generates that new potential chiral center at that group, and we're going to see that in a moment. And this is a much more stable structure compared to the linear form. Think of having a reactive group and a reactive group, and then just in solution interacting or potentially interacting and bumping into each other, forming this reaction. This makes those ring structures the more stable conformations. Looking at how that happens in solution, here to form that 6-member ring, we're going to see the hydroxyl at position 5, attacking the carbonyl group, forming that cyclicization. Now, like I said, this generates a new chiral center. This chiral center is going to produce another isomer. We call this an anomer. This is going to be labeled depending on where that hydroxyl is relative to the ring. If it's underneath the ring, we refer to it as an alpha isomer. If it's above the ring, we refer to it as a beta isomer. So now we're going to label these carbohydrate molecules based on the last chiral center in their linear form, L and D, the chiral center formed as a part of the cyclicization, alpha or beta, plus if it's going to produce a ring structure, a 5-member ring or a 6-member ring, or pyranose. So for example, here we have a beta D glucopyranose. It's a glucose ring forming a pyran ring. A glucose forming a pyran ring has a D form of glucose. It's in the beta conformation. You're not going to be expected to name. I'm not going to give you a sugar molecule and expect you to name it or identify what that is. But you do need to understand what these mean because it might come up. It's going to come up when we start thinking about glycosidic bonds. So understanding this alpha and beta conformation in this ring structure will be critical. And like I mentioned, that anomeric carbon, or that carbon that's generated from the cyclicization reaction, is also a chiral center. And we can see that using a Hayworth projection. So this is how we would draw a cyclic ring structure with those hydroxyl groups either shown below the ring or above the ring. We're always focusing on that anomeric carbon, and that anomeric carbon is often going to be either the C1 or the C2 carbon, depending if it's an aldose or a ketone. So here, if we label this 1, 2, 3, 4, 5, 6, that C1 carbon is right now in the alpha conformation. That hydroxyl group is below. On the right, we have that C1 carbon here with hydroxyl group above the ring in its beta conformation. So this is the intramolecular cyclicization reaction. However, this is not the only potential reaction that can occur within linear carbohydrates. It's most likely to occur because the hydroxyl groups and the carbonyl group are part of the same structure and they were going to react with each other. But that reactive carbonyl group means that there can be interactions with other molecules, reactions with other molecules, and we're going to see that in a moment. So these are very reactive groups, meaning that carbohydrates in their linear form are also very reactive. But all those other functional groups are also sites of potential modification and reactions as well. If you remember from DNA and RNA, so ribonucleic acid or deoxyribonucleic acid, deoxy means that one of the hydroxyl groups has been removed. So this is a modification that can happen to glucose or to carbohydrate molecules. We're also going to encounter phosphorylation reactions, so having phosphate groups added to glucose molecules. This is going to be part of the metabolic pathways as well. These modifications now add even more complexity to the potential structures and functional groups we're going to see on these carbohydrates. So there is some terminology that we've kind of alluded to or talked about that I just want to put more explicitly here on the slide. What are we referring to as an anomer, and what we'll see as an epimer, isomers, constitutional isomers? This will be important knowledge for you as well. So an isomer is just anything that has the same molecular formula but different structure. So for example, we see glucose and fructose. Both have the C6H12O6 molecular formula, but they're organized in very different ways. Constitutional isomers have a different order of the functional groups. So they're same general structure, but different order of those functional groups. And stereoisomers have the same formula and order, but they can be either non-superimposable mirror images, so in enantiomers, or a diastereomer where they're not going to be mirror images of each other. And different examples of a diastereomer can be an epimer, which is going to be different at one asymmetric carbon, or an anomer, where it's going to be different at a newly formed anomeric carbon within that ring structure. So beta and alpha glucose, D-glucose, those are examples of anomers. We're going to see other examples of potential epimers as well as we move on through our examination of carbohydrates. So, looking at glucose and galactose, which I've highlighted over here, what kind of isomers are glucose and galactose? You can choose all that apply. There's multiple correct answers here. Are they constitutional isomers, stereoisomers, enantiomers, diastereomers, epimers, anomers? What are they? So it looks like both three leading the pack. Stereoisomers, diastereomers, epimers seem to be the top choices, which are correct. So looking at constitutional isomers, thinking back here, different order of functional groups binding, looking at glucose and galactose. They have the same order, aldehyde at the top, hydroxyl group moving down that carbon backbone to eventually that terminal hydroxyl group. So it is the same order. So not a constitutional isomer. A stereoisomer, same formula and order. So they do have the same formula. They do have the same order of functional groups. That's right. Enantiomer, non-superimposable mirror images. Not really a non-superimposable mirror image. So looking at this, if we were to put an image together, they're not a non-superimposable mirror image of each other. So that would make diastereomers correct. Epimer, are they different at one chiral center? Yes. Are they an anomer? Is there different at a newly formed anomeric carbon? No, they're not in a ring structure. So these are stereoisomers that are also diastereomers that are also epimers. So all of these terminologies can apply to glucose and galactose in this comparison. So, but why? I've spent a lot of time talking about the organization of these functional groups and why we care, or why they're important. But I want you to put this into the context of an enzyme now trying to interact with these different molecules. Here's glucokinase. We're going to see glucokinase on Thursday as well, and its comparable protein hexokinase. This is a protein that catalyzes a phosphorylation reaction on glucose, producing glucose 6-phosphate. Now we see the ribbon structure here for this protein. We can also see the space filling model here on the right. When glucose binds to glucokinase, it undergoes a conformational change, folding around the glucose and allowing it to carry out this enzymatic reaction. Now, glucose binds within this active site of this protein, forming non-covalent interactions with the amino acids within that active site. Now, these interactions are very important for glucose binding. All of those functional groups on glucose, those hydroxyl groups, form these hydrogen bonds or these interactions with these amino acid side chains. The Km for glucose is about 6 to 10 millimolar. So the Km of this protein, glucokinase for glucose, is 6 to 10 millimolar. Remember Km, the Michaelis constant. This is not exactly the same as the dissociation constant, but gives a strong indication for the preferential substrates. Looking at galactose, that only has one difference in the position. Same chemical formula, generally the same structure, just one of those hydroxyl groups is in a different conformation. If we were to put galactose in this hydroxyl group, that hydroxyl group would be pointing up, away from some of these amino acids. This would weaken the interaction. And what we see within this enzyme is that it does not have a measurable Km for galactose, meaning it cannot bind galactose and it cannot carry out an enzymatic reaction on galactose because of this one change in the conformation of this protein. This is how our enzymes carry out specific reactions in specific molecules. By where those functional groups are associated within that active site, this can impact how it binds and the enzymatic reaction that it can carry out. So even though these are very similar proteins, they're epimers of each other, or very similar carbohydrates, they're epimers of each other, they are not the same substrate for these enzymes. And galactose cannot be used in place of glucose, even though it's a very similar carbohydrate. Now, I also mentioned that that carbonyl group is a very reactive group. Because it's so reactive, it's this kind of reactive center of this carbohydrate. This carbohydrate can actually move back and forth through that linear form. Once it's formed that cyclic structure, even though that is a very stable structure, it can become its linear structure again and fold into a different cyclic structure afterwards. So it's constantly in equilibrium within solution. This means that that carbonyl group has the potential to not only react with itself, but also react with other molecules in solution or in the environment within the cell. We call these molecules that have the potential to react with other molecules reducing sugars, because they can carry out an oxidation reduction reaction with another molecule, becoming oxidized and reducing that other molecule as well. So this is a reducing sugar that has a free carbonyl group available to it. We can do a test using copper to determine if these are present in solution. So in this test, copper becomes reduced, that carbohydrate becomes oxidized, and we can detect that as well. The reason I bring this up is because this linear form becomes very important when we start thinking about carbohydrate structure later on, but also when we start thinking later on about hemoglobin. And remember that case study? We're going to come back to it in a moment. But first, I want you to look at these structures and determine which of them can be considered reducing sugars. Which of the following are going to be reducing sugars? Looking at the cyclic structures of all these carbohydrates, which have the potential to open back up into the linear form and are considered a reducing sugar? I'll give you a hint. There's two possible correct answers. Looks like the majority are going with the top two, which is correct. So the top two are reducing sugars. Now let's look at why. What about them made them reducing sugars? Anyone want to answer? I got a mic. What made those two reducing sugars?
The free OH group on their link. I don't know.
Yep. The free hydroxyl group on that anomeric carbon. So to identify a reducing sugar, the first step is always to identify that anomeric carbon. So looking for that O8, that oxygen within the ring, it's going to be often the one next to it. The way we usually draw it is to the right. So 1, 2, 3, 4, 5, 6. The anomeric carbon here. And then having that free OH group. So here's the anomeric carbon, free OH group. Looking at this anomeric carbon, it does have an oxygen, but that's part of another interacting with another molecule. This is a glycosidic bond. So there's no free OH group here. Likewise for ATP, the anomeric carbon is participating in a bond now with something else. There's no free hydroxyl group. So this is not going to be a reducing sugar, which is very useful. We don't want our ATP falling apart as well within our DNA. So it's a very stable structure. So now coming back to hemoglobin, that hemoglobin A1C is an example of a glycation reaction that occurs on hemoglobin. This is that carbonyl group on that carbohydrate reacting with the N-terminus of that hemoglobin, forming a very strong reaction, forming a really strong reaction. We call this glycation. Now, this is not to be confused with glycosylation. Glycosylation is an enzymatic reaction where a protein adds a glucose molecule onto another protein, an amino acid side chains. This is a non-enzymatic reaction, so we call this glycation. Now, this is important when we think about hemoglobin, because hemoglobins are in red blood cells. Red blood cells do not have a nucleus. They last about 90 to 120 days. But because they don't have a nucleus, they're not undergoing gene expression. They're not producing any new protein. So all the hemoglobin within that red blood cells had to be synthesized, had to be translated when that cell divided and lost its nucleus. So the typical lifespan of 90 to 120 days is also the typical lifespan of a hemoglobin molecule. We can look at how glycated those hemoglobin molecules are to see how much glucose they've been exposed to over that 90 to 120 days. The more glucose that's present within the blood, within the red blood cells, the more glycation that's going to occur. So this becomes a proxy for us, or for individuals and physicians, for the blood glucose levels of an individual over a long period of time, over that 90 to 120 days or 3 to 4 months. The typical ranges for blood glucose levels are about 6%. So hemoglobin A1C level, the typical range for an individual is less than 6%. So 6% of your hemoglobin being glycated is in the normal range, or less than 6%. Someone who has prediabetes is going to be somewhere between 6 to 6.4%. Someone who has a diagnosis of diabetes is often going to be greater than 6.5%. So more than 6.5% of their hemoglobin is glycated during an individual single test. So this is a really useful product, but it all comes down to how those glucose molecules are interacting and reacting with the proteins and functional groups around them. So coming back to our case study, looking at RP, RP had a hemoglobin A1C level of 12.1%. That indicates that 12.1% of their hemoglobin was glycated. This indicates very high blood glucose levels over those 3 to 4 months, causing the glycation of that hemoglobin molecule. Now, this has no impact on hemoglobin function. This is more just, I think I heard one example calling it just frosting, that's added on top of, onto each one of these hemoglobin molecules. So it's not going to impact their function, but we can use this as a diagnostic tool. Part of the reason why individuals are able to maintain or keep that glycation level low at around that 6% or below that 6% is how our body is able to regulate our blood glucose levels. And we're going to talk about that when we start thinking about protein transport at the end of this lecture. So just in summary, thinking about carbohydrates or some of these simple monosaccharides we've explored. Monosaccharides are going to be the simplest form. They can be a triose, a tetrose, pentose, hexose, even a septose, and even higher. They all have the general formula of a carbon or hydrated carbon, CH2O. Carbohydrates are either going to be an aldose or ketose, depending on where that carbonyl group is present. There's lots of chiral structures or chiral centers within those carbohydrates, which increase the complexity and the number of potential isomers, again, increasing the complexity and the diversity of carbohydrate structures as well, even if they have the same molecular formula. We will define them or identify them as D, capital L, based on the chiral center furthest away from that carbonyl group. Biologically for us, D sugars or D carbohydrates are important. These are the ones that our enzymes have evolved to use, just like L amino acids or what our enzymes have evolved to use as well. Our enzymes have evolved to use D carbohydrates. That cyclicalization reaction results in 2 more structures at the anomeric carbon, and this can be in the alpha or the beta conformation, and knowing the difference between those, and I have it highlighted here, will be important for the second half of this lecture. And then reducing sugars are sugars that have that free carbonyl group that can undergo react with other molecules. Either they undergo a cyclicalization reaction with an intramolecular reaction, or an intermolecular reaction, reacting with other molecules. Yeah, we're going to take a 10-minute break. I think we'll be okay until 10:08. Then we'll come back. I'll answer any questions here at the front during the break as well. It's more about where this group is. So this reaction is formed based on the hydroxyl group. So this is what used to be the permanent. So we're trying to find something that used to have that. So okay, what's your question? It's more about its position of this. So that ring. Go back. Go back. When that ring forms, that pushes it back up, forming that new hydroxyl group. So that hydroxyl group is formed from that reaction. So that hydroxyl group, the one that's next to that O in the ring, is what used to be carbon. Again, that hydroxyl group that's next to the O is what used to be the carbon. So as long as that hydroxyl group is still there, it can become a carbon. No, it could be C2. In a ketose, it could be C2, and an aldose is C2. Yeah, so when we organize it in this linear form, is how we count it. starting from the carbonyl group, putting the carbonyl group at the top, and starting to count down. So here, the carbonyl group is at the top, and the ketones, the carbonyl group is one down. Yeah. No, they're going to be on different sides. And it's more going to be their orientation relative. So this might be up, that might be down, this one might be up and down. So when you switch them, now they're going to be opposite. It's still true, but when you flip them over, it's going to become the same. You can kind of look at the separation. All of them together means that when you flip one, it changes the way things evolve. Not necessarily. So you might have a molecule that could have the same formula, but the way they're organized, you might not have an aldehyde, you have a ketone. It's not going to have multiple aldehydes. It's not multiple aldehydes, so it's not a single sugar, but all sugars will have that. Yep, the C1 has to be the one that used to be C1 or C2 is the one that used to be the carbon. So that used to be the carbon. Because that one doesn't have a free hydroxyl group, it has never had potential for carbon. Okay, has to be. Yeah, usually we got in this orientation, but there will be some molecules. You're going to see an example later on where it might be flipped over, and it takes a little bit more thought process. I'll show you the process. I think I replied. Yeah. Thanks for the invite. I think I said like another event in the future, probably. Yep. No, that's that oxygen is now part of the second part. Reforming the carbonyl group and then breaking it apart. So this, even though this has a hydroxy. We will come back. All right, we will come back. And we're going to continue our discussion around glycosidic bonds. But I did get a number of questions about this Menti. So I do want to just answer or kind of give you a quick way to be able to identify, to help you to identify where that anomeric carbon is. Looking at the cyclicalization reaction, that hydroxyl group is going to attack that carbonyl group, forming the ring. So that hydroxyl group is now the oxygen, what was that hydroxyl group is now the oxygen that is in that ring. So being able to find that hydroxyl group is important for that ring, but that new hydroxyl group is what's left of that carbonyl. So when you're trying to identify which ones can open back up, you need to find that free hydroxyl group that's next to that oxygen in the ring. That free hydroxyl group can then have a reaction where it opens back up to regenerate the linear form. So identify that oxygen in the ring, look on either side of it to see which one has a hydroxyl group, and that's going to be either C1 if it's an aldose or C2 if it's a ketose, and that's where you're looking for that structure that's going to open itself back up. Now this is important when we start thinking about what happens at this anomeric carbon when it starts to form more complex carbohydrates, disaccharides, polysaccharides, oligosaccharides, as they start to form and react together. These reactions that occur at these anomeric carbons are condensation reactions or hydrolysis reactions when they break apart, and these form glycosidic bonds. These are bonds that are going to connect the anomeric carbon to another molecule. This is how carbohydrates get linked together in more complex structures. That condensation reaction, them coming together, we call it a condensation reaction because it's going to generate water, condense two molecules into one, or a hydrolysis reaction where water comes back in and breaks it apart. We join these monosaccharides together, or our enzymes join monosaccharides together into glycosidic linkages or glycosidic bonds to form disaccharides and then these more complex structures. The naming convention is important. Looking at the structure of glucose and fructose coming together, looking for the labeling, we're seeing that oxygen there. We're finding the one that has the hydroxyl group. That's going to be C1, 2, 3, 4, 5, 6. Looking on fructose, we're looking at that oxygen. Now we're looking for the one that has the hydroxyl on it. This one doesn't. So this is going to be C1, C2, 3, 4, 5, 6. So this is going to be linking C1 to C2. And we label this as a 1 to 2 glycosidic linkage. And in particular, this is alpha. So that hydroxyl group is in an alpha conformation. So this is an alpha 1,2 glycosidic bond. The orientation of this bond is important for how our enzymes are able to put them together, but also potentially break them apart. And we're going to see that having specific enzymes to break these specific bonds is critical to how we digest and consume and digest carbohydrates in order to derive energy from them. So again, variations of the glycosidic bond are going to be dependent on where the anomeric carbon is or where the hydroxyl group is. Here's looking at the formation of methyl D glycoside or methyl D glucose. That methyl hydroxide is going to be able to react with this group, with this glucose group, forming either the alpha or the beta orientation. If it attacks from the top, it's going to form the beta conformation. If it attacks from the bottom, it's going to be in the alpha conformation. So that position relative to the ring is going to dictate whether it's in this alpha conformation below the ring or beta conformation above the ring. So this is really important when we start thinking about the more 3D structure of these carbohydrates and how they interact with the functional groups within our functional groups within our enzymes. We can also have other variations of glycosidic bonds. We've seen mostly O-linked, where we looked at the example so far. We're connecting these monosaccharides together as disaccharides. So we're looking at hydroxyl groups reacting to another hydroxyl group. But other nucleophiles can also attack these groups, forming different types of glycosidic bonds. In particular, an example you're going to see is an N-link group, where an amino group or amine group is going to attack that anomeric carbon, forming this glycosidic bond. An example will be within DNA. DNA has an N-glycosidic bond. All those nucleotides have an N-glycosidic bond on the ribose and that nucleotide base, connecting them together. We also have N-linked glycosidic bonds within our proteins, within a post-translational modification that occurs on our amino acids. So asparagine is an example of of functional groups on an amino acid that contains an amino group or a nitrogen group that can attack that anomeric carbon and become an N-linked glycosylation. We call these glycoproteins, or proteins that have a glucose attached to them as that post-translational modification. We also have O-linked examples, so serine and threonine. So these are going to be hydroxyl groups that can attack that anomeric carbon, forming an O-linked glycosylation and glycosidic bond. All of these are different examples of post-translational modifications that can occur. But because protein or carbohydrates can be linked together in long chains, that may not only be a single glucose molecule attached. This may now allows this hydroxyl group to potentially react with another anomeric carbon, forming long chains of carbohydrates on these proteins. And we call these glycans or glycated glycoproteins, and these may be on the surface of a protein forming some sort of a recognition site. Because these carbohydrates can react with one another, they can also start to form very long chains. So we call these complex carbohydrates. We've looked at monosaccharides so far, so only one carbohydrate, a disaccharide having two carbohydrates. But these carbohydrates can form these polymers. So oligosaccharides that have somewhere between 3 to 20, or a polysaccharide that can have up to thousands. Like polymers of DNA, or DNA bases, is a polymer, polynucleotide, DNAs of amino acids, polypeptides, we can have polysaccharides as well. So this is another polymer within a cellular context. Some examples we're going to explore a little bit more, we've seen already. So we've seen sucrose, and then alpha-1,2 linkage. So every sucrose molecule is going to have this alpha-1,2 linkage. This is what sucrose is, a glucose and fructose molecule connected with that alpha-1,2 linkage. This is a disaccharide because that's two carbohydrates. Lactose is a galactose and glucose connected with a beta-1,4 linkage. So every lactose molecule will have a galactose carbohydrate, glucose carbohydrate, linked in this exact same glycosidic bond. Amylose is what you're normally going to see in starch. So amylose is going to be two glucose molecules linked in an alpha-1,4 linkage. So looking at the orientation, we can see here's the oxygen, looking for the one that has the hydroxide group. That's 1, 2, 3, 4, 5, 6. On this side, 1, 2, 3, 4, 5, 6. We see a 1,4 linkage. It's below the ring, so it's an alpha conformation. It's an alpha-1,4 linkage. Amylopectin is just a branched form of starch. So this is going to be the same glucose molecules linked together in not just alpha-1,4 linkages, but also having alpha-1,6 branch points. So again, looking at this, 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So that's a 1 to 6 linkage, and it's an alpha conformation. Because of all these potential linkages and these branching points within these molecules, this, again, I'm going to emphasize the point, increases the complexity of carbohydrates. So these polysaccharides now can form recognition sequences on the surface of cells. These very large polymers can be attached to proteins on the surface, forming these recognition sites, allowing a cell to identify itself versus a potential foreign invader, initiating an immune response. It can also be used for energy storage, so how we're going to see how plants might store glucose and carbohydrates as starch. We're going to store them in our bodies as glycogen. And as glycogen, this is what we call a homopolymer, so it's only glucose molecules stored together, or homoglycan, and it's consisting of the exact same monosaccharide. But we can also have the heteropolymers that we see on the surface of cells. It might be a glucose, fructose, galactose, ribose, linked together, forming this specific sequence or orientation of carbohydrates on the surface of the cell. This is very important, as I mentioned, for recognition, for proteins to be able to recognize other proteins or self-proteins, and for potential immune response as well. So looking at some of these complex carbohydrates, let's start with starch. Starch, what we get in your food, if you like potatoes, like French fries, like anything that might be sugar or sweet, any baked goods, you're going to see a lot of amylose in there, because that is what starch is. These are just unbranched glucose units linked together in an alpha-1,4 linkage. A more complex starch might be amylopectin, where it's not only alpha-1,4 linkages, but also those alpha-1,6 branch points. And this occurs roughly every 30 glucose units. This is going to be different from the other storage unit we see in humans of glycogen, which has much more branching as well, making it take up a little bit more space. In order to break this down for our cells, in order to break this down and be able to derive energy from it, we need enzymes to break down this into its constituent monosaccharides. Our body cannot absorb from our digestive tract disaccharides or polysaccharides, and we can only absorb single molecules or monomers of these polysaccharides. So glucose, galactose, fructose, we're not going to be absorbing amylose. So we need enzymes that break them down. So alpha amylase is secreted by both our salivary glands and our pancreas and helps to break down these complex starches into simple monosaccharides that we can absorb. Looking at the structure of amylopectin, because every single one of these residues has the same linkage, it's a glucose linked in an alpha-1,4 linkage as a polymer, that amylase is able to bind to any region within the structure and start to break it down into smaller oligosaccharides and disaccharides. In particular, maltose, which is 2 glucose molecules, isomaltose, which is 2 glucose molecules now in that 1,6 linkage, or you're going to see some limit dextrins, which are much larger complexes that may not fit within the active site of amylase anymore. But as we start to break them down into smaller and smaller molecules, this gives now our body a chance to bring in other enzymes that can break it apart into its constituent monosaccharides and absorb them. So I mentioned that amylase is produced by both the salivary glands and the pancreas. Why is it that we need to produce it by 2 different points within our digestive system? So those who have taken physiology, you might know this, but again, connecting to some of our previous discussions as well. Is it that the salivary glands are small and don't secrete enough of the enzyme? Salivary amylase is degraded by the time it reaches the intestine, so more is needed. The pancreatic amylase has a higher Kcat, where pancreatic amylase releases insulin. The majority going with B, absolutely. So thinking about our digestive system, what's happening in our digestive system, saliva is releasing amylase, beginning the process of breaking down carbohydrates in the food we ingest in our saliva. As it moves down through our digestive system into our stomach, pH decreases dramatically, causes the protein to be denatured. The amylase itself is degraded and reabsorbed. A lot of the protein that you consume or you absorb in your digestive tract is your own proteins that you're releasing into your digestive tract as well. They get broken down and reabsorbed. So the salivary amylase gets degraded, digested, reabsorbed as amino acids, and the pancreas releases another version of amylase that continues the process of carbohydrate breakdown until we can actually absorb it. So looking at another complex carbohydrate, cellulose. This is one of the most abundant carbohydrates, organic compounds, and serves mostly a structural role in plants. It'll come back, I hope. It's also made of glucose. So like starch, like amylose, like amylopectin, is also just glucose molecules linked together in a chain. The difference being that it's a beta-1,4 linkage, not an alpha-1,4, a beta-1,4. So the orientation of the hydroxyl group now above the ring causes a different conformation, a very much more linear conformation of the cellulose. We do not have an enzyme that can break down glucose in this conformation. We do not have an enzyme that has an active site that can fit cellulose to break apart this chain. So this means that cellulose moves through our digestive system without any enzyme able to do anything and becomes what we call fiber, becomes bulk, enters into our large intestine, and we excrete it in our feces. This linear conformation of cellulose is really important for plants because it gives it a structural conformation, a structural feature. It allows it to form this long chain or mesh-like feature on the surface of the cells, giving it a structure. This also gives it a negative iodine test. You may see starches or see carbohydrates referred to as either being a blue starch or a red starch, or blue on an iodine test or red on an iodine test. Something that's blue is a positive test on an iodine test, and that means that iodine can form a complex with it. So here is looking at this is amylose. So that glucose molecules connected in alpha-1,4 linkages. If you zoom in or if you look over here, you'll see this alpha-1,4 linkage over here. This causes amylose to form this kind of cyclic helical structure that iodine actually fits in really well and binds to and can associate with. This allows it to bind to the starch and turn it blue. Iodine is normally going to be very purpley blue, so this gives it a blue test. Now cellulose, because it's very linear, because of the structure of that, or the orientation of that bond, is a very linear structure and does not associate with iodine. So it becomes negative on an iodine test because of that structure of those molecules and how they're linked together. The last complex carbohydrate, I believe the last one we're going to talk about, is going to be glycogen. Now, glycogen is how we store glucose within our cells, particularly in the liver and muscle cell. So we can store fats for energy, we can store carbohydrates for energy too, in a small amount as glycogen. Typically, the amount of glycogen a human will have is probably enough to last about 24 to 48 hours if you're well-fed. If you had lots of carbohydrates the night before or over the period of the last week, you probably have about 24 to 48 hours of carbohydrates stored. Whereas fats, you can have weeks of fat stored to supply energy for some of those critical tissues. Like amylose or amylopectin, This is going to be glucose polymer, so a homoglycan, a homopolymer, linked together by alpha-1,4 linkages. And then we do also have an alpha-1,6 linkage, like amylopectin, except this occurs much more frequently, about once every 8 to 12 glucose units. So this becomes a chain or branching chain or a network of glucose molecules that we can store and keep within our cells to use as energy. At the center is a glycogenin protein, a dimer of glycogenin. And this is important because it's going to bind to the reducing ends. So remember, those reducing ends are very reactive. If the reducing end was exposed, it has the potential to react or become a linear form, start to react with other molecules. So here, this is linked together to a glycogenin molecule, glycosylation reaction that links them together and starts to build this chain from that protein at the center. Think of it like a seed that everything's now growing off of. This means that the glucose units get added from the non-reducing ends. So you're going to add glucose and build it up out from the glycogen molecule from the non-reducing ends. And then you're going to remove those glucose units one at a time as well. So you can think of it as either extending out from all of these or reducing in and removing glucose units one at a time. And when the glucose units gets removed, now it can be used for energy by that cell or by other cells within the body. So looking at the structure again, emphasizing the alpha-1,4 bond linking glucose molecules together, or that alpha-1,6 residue causing the branching points. And the branching allows lots of glucose molecules to be linked together in a relatively small space. Remember that reducing end is going to be the point of the molecule that can reform that linear structure or that carbonyl group. In order to prevent that happening, glycogenin is going to bind to that reducing end, keeping it stable. And then these non-reducing ends are going to be where you can cleave off glucose molecules to remove them to allow that cell to use energy at a later point. So thinking about digestion now and the use and how we transport and use them in the body, those glucose molecules have to undergo digestion in order to be absorbed as those monosaccharides. As we mentioned, starting within the mouth, salivary amylase will start to break down those carbohydrates. That carbohydrate digestion is going to be completed within the small intestine, breaking down those complex carbohydrates to galactose, fructose, and glucose, where they can be absorbed by glucose transporters within the small intestine, make it into the bloodstream. The enzymes that break them down to those single monosaccharides are going to be lactase, maltase, and sucrase. Based on the name, lactase is going to break down lactose from galactose and glucose down to into galactose and glucose. Maltase breaks down maltose into 2 glucose units. Sucrase breaks down sucrose into glucose and fructose. These enzymes are found attached to the cells in the microvilli in the small intestine. So as those disaccharides are moving through the small intestine, they're interacting with those enzymes, breaking them down, allowing them to be absorbed. It's all mediated by the proteins here that are present on the surface of these cells. These proteins have to have specific recognition sites that allow them to interact with those substrates. Lactase is not going to be able to break down maltose because of the orientation of those functional groups within the active site. Only lactose is going to fit correctly, allowing it to break that beta-1,4 bond and have the hydroxyl groups at the specific orientation to interact. So it's all about those amino acid side chains that are going to be present within the active site interacting with those substrates. So looking at maltose, if we go through this example of numbering maltose and seeing how it's again broken down, so the 2 glucose units. Again, I'm going to emphasize this point, being able to identify and number these carbons. That oxygen within the ring, looking for the group next to it that has hydroxyl group. So it's going to be 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. This is a 1,4 bond. It's in the alpha position. So maltase is going to cleave that alpha 1,4 bond, generating those 2 glucose molecules. Looking at lactose, and lactose is a little bit of a trickier example, but again, following that strategy, you can label it correctly. Find that oxygen in the ring, look for the one next to it that had a hydroxyl group. 1, 2, 3, 4, 5, 6. Now looking at the other side, oxygen in the ring is a different orientation than we normally see, but look for the one next to it that has a hydroxyl group. This one doesn't, so we're going to label this counterclockwise. 1, 2, 3, 4, 5, 6. So this is also a 1,4 linkage. But now that oxygen is going to be in the beta position, so this is a beta-1,4 bond. So lactase is going to cleave the beta-1,4 bond, producing galactose and glucose. But remember we said that cellulose also has a beta-1,4 bond. But cellulose, or lactase, cannot break down cellulose, the cellular bios. Even though it's a beta-1,4 bond, lactose is galactose and glucose, so the orientation of those hydroxyl groups on the galactose molecule is specific, where cellulose is glucose and glucose. So the orientation is really important and critical for how that enzyme is able to interact with that molecule. So we cannot break down cellulose, even though we have an enzyme that breaks beta-1,4 bonds, because of the specific orientation of those hydroxyl groups on the substrate. So looking at this, asking you a question now. Can alpha amylase, which breaks an alpha-1,4 bond, hydrolyze the glycosidic bond in the structure below? Yes or no? Looking at the responses coming in, say about 60-40, 70-30 split? No? Yeah, 70-30 split maybe? Yeah, 70-30 split is saying no, some people saying yes. The correct response here would be no. So let's take a look at that. Looking at the structure again, I feel like I'm on repeat, but numbering, find that oxygen in the ring. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So it is a 1,4 bond, but looking at where that hydroxyl is relative to the ring, that bond is coming above the ring, so this is a beta-1,4 bond. Amylase breaks alpha-1,4 bonds, not beta-1,4 bonds, so this one, amylase is unable to break this bond here, and this is actually cellulose's example as well. So remember that orientation, alpha below, beta above the ring. So it's not just about numbering, but then correctly identifying the orientation of that structure as well. I have another question for you now. So we have lots of techniques to sequence polymers. DNA sequence or Sanger sequencing allows us to sequence or identify the order of polymers within a DNA chain, a polynucleotide. We looked at protein sequencing in this course, Edman degradation, mass spec to an extent as well, allowing us to identify the order of amino acids in a polypeptide. Now the other polymer we've just looked at is carbohydrates or polysaccharides. But sugar sequencing, while possible, is very difficult. Why do you think that is the case? So looking at some of the responses, let's see. Isomers chirality, absolutely. Isomers, let's see, I'll eat it all, yes. Isomers, 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 isomers. DNA has 4, proteins have 20, sugars have so many more, absolutely. So I want to highlight this one because this is a great example. Thinking about a polynucleotide, so like DNA, at each position, there's only 4 possible bases, A, C, G, T. In a protein, there are 20 possibilities at each position, any of the 20 amino acids. For a carbohydrate, say a hexose, 16 possible isomers that all have the exact same conformation. And then the next position might not be a hexose, might be a pentose, might be a different structure. So it becomes hundreds, maybe even hundreds of possibilities at each position. On top of that, remember when we did mass spec and we talked about isoleucine and leucine, how it was very difficult for mass spec to be able to identify the difference between them because they have the same atomic weight? We have the same challenge here with carbohydrates. Glucose and galactose have the exact same molecular structure and the same atomic weight. It's very difficult to tell the difference between those two molecules on mass spec. So while sugar sequencing is possible, and there are some techniques to do it, involving enzymatic reactions, modifications, and mass spec as well, it is very difficult. There's differences in composition, differences in the way they're connected, differences in the configuration, and all of these make it very difficult to identify the sequence of a glycan on the surface of a protein or within a more complex carbohydrate if it's not a homopolymer. So while it's possible, it is very complicated. Now coming back to our underlying theme of digestion and moving of glucose within the body, we need to see what happens to glucose as it moves from our digestive tract into our bloodstream to be able to use by our cells. And this is critical, because remember I mentioned at the top of the class, our brain requires glucose for energy. It needs about 130 grams per day just to maintain brain activity and brain function. And glucose, however, though, is a very polar molecule. All those hydroxyl groups, all those polar atoms on that molecule mean it can't cross the plasma membrane. It needs proteins to help facilitate this movement across these cells and into all these different tissues. We looked at the glucose 1 transporter earlier in the course, so I'm going to bring it back up again, and we're going to look at all the different glucose transporters that help facilitate this movement. These transporters help move glucose along its concentration gradient, so from high concentration to low concentration. But also we have transporters that move it against its concentration gradient using secondary active transport. So the movement becomes critical in order to fuel our cells and cellular activities. Now, different cells are going to take advantage of different transporters based on how they utilize glucose and based on the cellular conditions in order to maintain homeostasis. So the three examples we're going to see commonly are sodium-glucose cotransporters. So these are going to be the secondary active transports, taking advantage of that sodium-potassium concentration gradient in the cell to allow sodium to come with its concentration gradient and glucose to come against its concentration gradient. We also have facilitated diffusion, so glucose transporters, and fructose transporters as well. Remember, the chain difference in that orientation and structure of glucose and fructose means that we need to have a different protein to be able to effectively move it across that plasma membrane. So fructose has its own transporter. So here's a selected list of glucose transporters. This isn't all of them. I think there's up to 12 or 13 different glucose transporters encoded within the human genome and expressed on different tissues. But I did want to highlight a few of the ones you're going to see more often in your studies and potentially in clinical practice if you do choose to go into a professional program. So SGLT1 and 2, these are going to be your secondary active transporters. These help move glucose against its concentration gradient, so from the small intestine into the cells of the lumen, or the cells surrounding the small intestine. And in your kidney, so filtering out glucose out of the kidney, so again, against its concentration gradient. Glucose transporter 1, which we saw earlier in the course, I get the example of protein structure. So this helps to move glucose with its concentration gradient, facilitate diffusion, and this helps get glucose into red blood cells, but also across the blood-brain barrier. Glucose transporter 2, you also see this in the small intestine. So again, moving now from inside the cells surrounding the small intestine into the blood. You also see this in the liver and the pancreas. Glucose transporter 3 is facilitated diffusion, and you see this mostly in the neurons. Now I want to call your attention now to its Km. So these are transporters, they're not enzymes, but they do carry out a function. They move something from one side of the cell to the other side of the cell, so they do follow Michaelis-Menten kinetics. So we do assign a Km to these for glucose. Neurons, which have the highest need, this is your brain activity, for glucose, their glucose transporters have the lowest Km. So they can have that bind to glucose most efficiently and move it across that plasma membrane more quickly than any other cell type in order allowing them to use glucose effectively. Glucose transporter 4 is what you typically see on muscle and adipose tissue, and this allows glucose to get into these tissues, but it's insulin dependent. And we're going to see what that means in a second. And then glucose transporter 5 is what you see usually for fructose in order to get fructose into the small intestine and into the cells as well. There's no Km for glucose for the fructose transporter. It doesn't bind to glucose at all, so we can't assign a Km. I do want to highlight here that you do not need to memorize these Kms, but I would encourage you to memorize the glucose transporters, the cellular location, and any important facts like insulin dependence. So let's look at the movement of glucose through some of these cellular contexts. In the small intestine, in the lumen of the small intestine, you've just consumed a meal. All those enzymes and proteins are breaking down those polysaccharides into their constituent monosaccharides. These transporters are now going to move glucose from that relatively high concentration or potentially high concentration of glucose into the cellular space, its cytoplasm, where it's going to be a relatively low concentration. So after a meal, this is moving glucose along its concentration gradient. Now, from inside the cell that's lined the lumen of your small intestine, inside those cells will have a relatively higher concentration of glucose compared to your blood. So allowing glucose to move across this concentration gradient as well, now into the blood. So again, moving glucose from some place of high concentration to some place of relatively low concentration. Moving it into that bloodstream where now it can move to all your other cellular locations and tissues. Glucose transporter 4 is unique in that it's insulin dependent. It is not present at the surface of the cell of muscle and adipose tissue. Only when there's a relatively high level of glucose in the blood, triggering the pancreas to release insulin, that hormone, that insulin will bind to receptors on the surface of the muscle and adipose tissue and cause the movement of that transporter from vesicles inside the cell to the surface. This is like opening a door. Once at the surface, glucose now can move into those cells to be used for energy when there's relatively high amounts of glucose present within the cell. Now, this slide is not on your deck. I'm adding this for context only. The blood-brain barrier is just a unique place. Because of the tight junctions between those endothelial cells, between the bloodstream and the brain, in order for glucose to move and access those neurons, it needs to move through cells again. So we have glucose transporter 1 on the surface of those endothelial cells, allowing the glucose to move from the blood into the cells, and then from the cells back into the vicinity of those neurons. And those neurons can then use glucose transporter 3 to take that glucose into those cellular space. This is why the brain is such a unique place. Because of that surrounding endothelial space, everything has to move through cells in order to access or be accessible to those neurons. This is a part of the reason why the brain cannot use fatty acids for energy. Those fatty acids have to be moved through lipoproteins, and those lipoproteins cannot move through this blood-brain barrier. So the brain has to rely on glucose and these glucose transporters in order to access that energy. So let's look at what's happening between the fasting state and the fed state for blood glucose. During a fasting state, we have relatively low amounts of glucose. Typically, the blood concentration of glucose is somewhere between 4 to 5.5 millimolar. Looking at those KMs for those glucose transporters, we can start to see how glucose is going to move through these transporters, or how efficiently glucose will move through these transporters. Glucose transporter 1, which is on red blood cells in the blood-brain barrier, has a KM of about 6.9, so it's going to start to use some glucose as well. The concentration in the blood is going to be relatively close to the concentration, the KM for GLUT1. This is important because we need glucose to move from the blood into the blood-brain barrier, across the blood-brain barrier, but also for red blood cells. Red blood cells don't have mitochondria, and you can see later on, starting on Thursday and into next week, that the mitochondria allow cells to generate energy from other sources, including fatty acids. Because they don't have mitochondria, red blood cells rely exclusively on glucose for energy in order to maintain homeostasis. So these red blood cells need a steady flow of glucose in order to do their function and deliver oxygen to all our tissues. Glucose transporter 2, which typically we see on the liver and the pancreas. It has a KM of about 17.1, well above the concentration that you typically find in the fasting state within your red blood cells. That means that glucose is not going to move very effectively into your liver and into your pancreas during a fasted state. Remember, the pancreas is what's going to release insulin and glucagon in response to blood glucose levels. So during the state, glucagon is likely going to be released more, insulin won't. Your neurons, so once it's crossed the blood-brain barrier, the KM of neurons is about 1.4, well below the concentration typically seen in the blood. That means all that glucose will flow very freely into those neurons to be used for energy. Remember, neurons rely almost exclusively on glucose for energy. And muscle and adipose cells, because the pancreas is not releasing insulin, there's no glucose transporter present there, so they have no access to the glucose during that fasted state. All based on how these transporters are interacting with glucose and efficiently moving them across that plasma membrane. Looking during the fed state now, so you've just consumed a meal, maybe it's a high-carb meal, maybe it's just a regular meal with typically 40% carbohydrates. Looking at the concentration, blood glucose levels typically rise to about 6.5 to 11.1 millimolar in the fed state, which lasts usually about 2 to 4 hours after a meal. This is well above the KM for GLUT1. So red blood cells, the blood-brain barrier, are going to be getting lots of glucose. GLUT2, you're going to start to see glucose move into the pancreas. That pancreas now is going to be able to release insulin into the bloodstream. So now you have insulin flowing into your bloodstream. GLUT3, which the neurons, still very high, getting lots of glucose going into your brain after a meal. But that insulin now that's being released can bind to receptors on those adipose tissues and muscle tissues, the insulin receptors, cause glucose transporter 4 to move to the surface. And now these muscle cells can start to use glucose. Adipose tissue can start to store glucose as fats as well for energy. So this concentration and maintaining this blood glucose level allows the flow of glucose into tissues based on their need and based on the fuel that's being available at any given time. So coming back to our case study then, if you recall, RP was a 12-year-old who was recently diagnosed with type 1 diabetes mellitus, experienced recent fainting episodes following insulin injections, suggesting periods of hypoglycemia or low blood glucose following treatment. Why could insulin be leading to, why could insulin lead to hypoglycemia and fainting? Let me just get the Menti set up, and I'll read the responses. Okay, well, yep. So is it that insulin blocks GLU3, preventing neurons from getting glucose? Insulin and glucose are co-transported into cells using the SGLT1 transporter. Insulin activates GLUT1, causing red blood cells to use glucose for energy. Or insulin stimulates GLUT4, increasing the uptake of glucose by muscle and adipose cells. The vast majority are going with option D, which is correct. So it's all about that insulin sensitivity. So looking at what's happening. In a fasting state, where insulin now is present, we see that transporter now has moved to the surface of the glucose 4. The Km of GLUT4 transporter, 4.6, is very similar to the blood glucose levels. That means lots of glucose will be able to flow into those muscle cells continuously, limiting the amount of glucose available for neurons and red blood cells. So they're now competing with each other for the same glucose stores. So if you have a very low blood glucose level or a low blood glucose level, you get insulin, it's going to tank that blood glucose level. And anywhere below about, I think it's about 3.6, your brain doesn't get enough glucose to function. You start to get neurons start not being able to fire effectively, fainting, headache, dizziness, all those symptoms related to the decreased neural activity resulting from hypoglycemia. So all coming down to the movement of glucose and how these proteins contribute to the movement of glucose and maintaining blood glucose levels. So keep that in mind as we go onto Thursday, we start thinking about glycolysis and metabolism. And keep these in mind when we start thinking about the structure of carbohydrates, the differences between monosaccharides and polysaccharides, and the glycosidic bonds linking them together, and how glucose is transported between these different tissues, and how that contributes to how cells maintain homeostasis. With that, see you on Thursday. Enjoy the rest of your day. We only assign alpha and beta to the So if you look at this one, this is also beta because the C1 part is yeah, so thing that what happens in your center is getting a signal. So because it's just staying in the body, it's keep repeating until it's happening. So it's just like hit, hit, hit. It's like one hit, maybe it's for the mind higher, signals. But you can't have it. Because after it's a little bit, to give the perception sweetness without any activity. But it also means like when you think about something that the signaling cascade